Hello everyone, my name is Jwalanti Sundaram and I would like to explain something that is called bioaugmentation. So what exactly is bioaugmentation? It has been defined as introducing cultured microorganisms into contaminated environments to enhance bioremediation of pollutants. So to understand bioaugmentation, let me tell you what is bioremediation. So, bioremediation is the process of degrading pollutants or certain chemicals by living entities. So, these could be plants, microbes, fungi, any living entity. When it degrades a pollutant, the term is called bioremediation. So, what is bioaugmentation exactly? You are introducing cultured microorganisms. So, when I say cultured, I don't mean that these microbes wear British top hats. No, they've been grown in labs and can be modified. They have been grown in labs, they've been tested, and then they're introducing them into contaminated environments so that they help in bioremediation. Now let's look at this diagram to understand the concept better. So we know that pollutants enter the environment through, or they enter nature through various sources. So they could be oil or toxic spills in the ocean, sewage, factory waste, all of the runoff will eventually result in the pollutant entering either soil or water. Now both these environments are already known to have microorganisms which will start the process of bioremediation and will start degrading the pollutants. So what is bioaugmentation is, along with these already present microorganisms, you are going to add certain additional microbes. So on the right hand side you can see that we are introducing cultured microbial consortia, where augmenting or adding them to this particular contaminated environment so that bioremediation can be better and it can be faster. Now here I would also like to add that in our case of bioaugmentation, we're providing the existing microbes with more soldiers. So we're giving them more, you could say, we're giving them more soldiers to fight the pollutant. But there's another concept called biostimulation, where instead of giving them soldiers, you actually give them more nutrients. So you give them more resources so that they can use these resources via biostimulation. They'll use the resources, they'll increase, they'll grow, and they will degrade the pollutant as well. So one example of using bioaugmentation is to treat industrial wastewater. Now, wastewater itself has microbes that can perform bioremediation, but the pollutants are in a very high concentration and are resistant to the existing microbes, which is why bioaugmentation has been preferred. Another advantage of bioaugmentation in this situation is that bioaugmented bacteria, or the bacteria that you grow, they can be tailored and customized to target the organism against which, not the organism, sorry, the pollutant against which you want degradation to occur. So you can customize your bacteria so that it will attack or degrade the particular pollutant that is contaminating the environment in that set time. So the table here gives a list of pollutants along with the bacteria that can degrade it or are added to the environment to augment the degradation of these pollutants. So a wide variety. So we see anilines, phenols, quinoline, some dyes, and we see the wide range of microbes from Acinetobacter to Bacillus to Paracoccus. Many bacteria can be used as for bioaugmentation of pollutants. Now the figure here is more of a visual representation of how bioaugmentation can help. So as you can see, on the left hand side the water seems more dirty, and on the right hand side it seems more clear, and the difference between the two pictures is the time for which the treatment has been given. So we can see results after 2 weeks of how the water is actually clearing up because of bioaugmentation. However, no particular technique or process is immortal to limitations. So some of them have been listed here. So one is the high mortality of the inoculated strains. Now you are introducing cultured and very well groomed microorganisms into an environment that could probably have anything that could, harm it. You're introducing it into something contaminated that could potentially harm the microbe. So your microbes could often end up dying even before they can contribute to the augmentation process. Another problem is bacteriophage infections. So bacteriophages are viruses that infect bacteria, and they often result in the death of bacteria, and they could lead to decrease of the number of bacteria present, and of course reduce the bioremediation as a result. The third limitation is protozoan grazing. So, the environment that you are adding the bacteria to is not perfect. It's going to have viruses, and along with them, it could also have protozoa. And these protozoa, they are known to feed on bacteria, and they will increase in number, and they will outgrow the bacteria. And this has been termed as protozoan grazing. So, if your bacteria could become fodder to either viruses or protozoa, which could lead an increase in their number and decrease in the number of the bacteria that you've added. So these are some of the limitations that you could face during bioaugmentation process. These are the references that I have used in creating this presentation. A special shout out goes to biorender.com. I used it to create the illustration scene in the presentation. So that's all from me. Thank you so much for patiently listening to what I have to say. I hope you've understood the topic. I will be posting all the references in the description below. Thank you so much.